Garu has a keen interest in behavioral ecology, and she is particularly interested in understanding the human interface of wildlife. Garu has some understanding. So, Charu, the floor is yours and all the best. Particularly highlighted. Garu has Garu, Garu has Garu, Garu has Garu,
So, good morning everyone. My name is Charushri and I'll be presenting on
Garu has
intraspecific and interspecific aggression in rhesus macaque. So, basically, as human
Garu
population
has
have increased, there is a significant overlap between human and animal ranges. And such prolonged human presence have obviously made alterations in conditions under which animals make behavioral decisions. And primates being one of the residents of cities now, the behavioral shifts in their life history patterns can be seen by their daily activity, dietary preferences, and habitat use, and even social interactions. So, rhesus macaques are basically widely distributed primate species. They are in constant conflict with humans, and they are young-living species with multi-male, multi-female groups, which can range up to from 20 to 200 individuals. And some of the cost and benefits of group living are mentioned here. I would like to highlight two of the costs that animals, and especially rhesus macaque, bear because of this particular trait, that is competition for food and space and reduced individual reproductive success. To mitigate such cost of being in a group living condition, animals produce not produce animals have different affiliative and agonistic behaviors, which help them compete for vital resources like food, space, and mate. And here is this is where aggression becomes competing for vital. So I'll be looking at intraspecific aggression, which becomes important now. So it helps in maintaining social hierarchy and giving access to high quality food and even resting sites. And along with interspecific aggression, mainly competing for humans, which are mainly seen in anthropogenic areas, mostly in Himalayan region. And along with rhesus macaque are also getting into aggressive interaction and hunting storms, cats, and even people sometimes. So, even though a lot of studies have been done on aggressive behavior on rhesus macaque, there are two things that have to be seen that there is no study which looks into behavioral changes. There are two gradient of different habitats, and mostly the habitats are basically varying in the spectrum of exposure to humans. And another thing is that there is very less work that has been done on temporal changes in habitat. So there are very less human presence in animal temporal changes here. It is sure that there are very less work that has been done to changes. So my objectives are to highlight frequency, level of aggression, intensity, and some of the points that are intraspecific and interspecific aggression. I'll specify frequency. It is sure this study has been obtained as per science, which will give us a comparison, and I'll explain if there is a change in the behavioral pattern, a decadal change in the behavior of rhesus macaque. So I have chosen two studies as a decadal change in the behavior of Kali temple, Buddha temple, RWI campus and one in Lachivara. So the Kali temple is the place where monkeys are hiding in as the first and one in Lachivara. So the second side is Kali temple, which is a park like sloth as the first and one in Lachivara. Next, the second side is Kali temple comparatively less there. WI campus is more or less similar to one in Lachivara. And then there is next side is Lachiwala, which WI campus is more or less which is near the Haridwar. And then there is the exposure Lachiwala, which all the sites WI campus is more or less which is near the Haridwar. And then there is the landscape. They all Lachiwala patches. All the sites WI campus more or less which is near the Lachiwala has a few mango orchards. Then there is the landscape. They all Lachiwala patches. All the sites WI campus more or less which is near the Lachiwala has a few mango orchards. I've said then the landscape. They all Lachiwala patches. All the sites you can see that Kali temple has the highest near the Lachiwala has a few mango. There have been very less few number of people being present. You can see that Kali temple has the highest near the Lachiwala and there have been very less campus and Buddha temples. You can see that Kali temple has the highest near the Lachiwala and there have been very less most of the time less than 50% people. You can see that like the number of the highest rolling Chihuahua and there have been very most of the time there have been incidents in campus where you can see that like the number of the highest rolling Chihuahua and I've selected 4 groups from my study. There have been incidents in campus where you can see that like the number of the highest rolling Chihuahua and I've selected 4 groups from my study. My sample time in campus where you can see that like the number of the highest rolling Chihuahua I've selected sample source from my study. 300 my sample time in campus I've done instantaneous like the sample rolling Chihuahua I've selected sample source from my study. 5 minutes and for humans done instantaneous like the sample 8 and 6 hours of class I've selected sample source from my study. 5 minutes and for humans done instantaneous infants under 8 and 6 hours of class and 8 and 6 class of humans are done instantaneous. 5 minutes and for humans done instantaneous and for aggressive behavior and 8 and 6 class of humans are done instantaneous. 5 minutes and for humans done instantaneous type of aggressive behavior, intensity of aggression, factors and also type of aggressive behavior, intensity of aggression, different factors and low level of aggression. So NAGAX is basically intensity of expressive type of aggression, like open mouth, this is a low level of NAGAX is basically different type of expressive type of aggression, like charging, like open mouth, high level of aggression, intact aggression, has behaviors type of aggression, like charging. I've also done this high level classification for humans, intact aggression, raising hand aggression, charging. I've also done this high level classification for humans, intact aggression, raising hand and for causal I've also done high level classification for humans, types, and for causal for humans, hands body damage for cats, type and tone. I've done G test of independence and static eye damage for cats, type and tone, 5-year test. I've done G test of independence and community damage and discussion, type and trust decision, 5-year test. So here we can see G test of independence and community damage and discussion, type and trust decision, and 5-year test. So here we can see G test of independence and community damage and discussion compared to Buddha temple and campus. So here we can So my reasoning to G test and common specialty compared to botanical and campus. I've written values of G. So here percentage of provisional G test and common specialty. So basically value compared to botanical and campus. I've written values of so that only less percentage of campus G test and common specialty. So basically value compared to botanical and campus. Since there's only directly values of so that only less percentage of campus G test and common specialty. And so basically value compared to botanical and campus. Since there's ultra specific values of aggression across regions and you can see that across the sites compared to since there's ultra specific compared to aggression other across regions for in and you can see that across the sites that adult females it is since there's ultra specific compared to and other the reason being adult females show more aggression against females than the reason is and for adult males it's other the reason being going to level of show more aggression against females than the reason is and for adult across the sites mid level of the reason being going to level of show more aggression against females. This is mainly because across the sites mid level of and going to level of after mid level high level of aggression. This is mainly because sorry low level of aggression and high level is typically less after mid level probably because aggression mainly we consider it from an epidemic high level is typically less fever after mid level probably because aggression mainly we consider it from an epidemic I'm going to that detail fever after and across regions probably because aggression mainly adult females show high level of aggression I'm going to that detail fever after and male show probably because aggression mainly adult females show high level of mostly adult males going to that show aggression for dominance because low level of behaviors like females and open mostly adult males going to that sorry show aggression for dominance excel level of behaviors like using part and mostly adult females sorry as I mentioned previously still show excel level of behavior head using part and even in most other adult females sorry as I mentioned also still show like the juveniles and other females using part particular habits where adult females restrain infants from growing show like the juveniles and this is something that we as well as adult females restrain this is one of the situations where like the juveniles and this is something that we as well as adult females restrain this is one of the situations where like the juveniles and this is something that we as well as it is dominance then restrain this is one of the situations where like the intergroup aggression in my study which is dominance as then restrain this is one of the situations where intergroup aggression in my study and then proximate factors witnessed in agents were again threat is seen as the major and then factors witnessed in agents were again some of the threats are seen as the major and aggression then factors witnessed in happening very very often again some of the threats are seen as major and aggression then factors so happening very very going to interspecific aggression is seen as major the frequency of aggression across aggression so happening very here again across species the frequency of human initiated aggression is mentioned here that so happening except for Kali Chana where I have mentioned that human initiated aggression is mentioned here that and other except for Kali Chana human beings have adopted cats and adult exclusively solution human beings have so here temple and campus exclusively shows similar results of plantation extension. So here temple and campus exposure to light shows similar results of plantation extension. So here temple and campus exposure to light shows level of aggression in interspecific study. Uh, Again, the results are similar to intraspecific studies, where both human and interspecific showed middle level of aggression. Again, the results are similar to intraspecific humans specifically, where both human and interspecific show high level of aggression. Is to look at again, the results are similar to intraspecific humans specifically, where both human and interspecific show high level of aggression. Level of interspecific aggression, the result generates initiated by macaques, where both human and there is high level of aggression. Significant difference here, most of them show level of initiated by aggression, where both humans there is high level of aggression. Significant difference here, and with respect to humans, there is no particular change which I believe is because when it comes to and with respect to humans, there is no particular change more than like I believe is because significant when it comes to and with respect to humans, there is no particular change more than like I believe is of significant when it comes to and with respect to humans. Atomic damage has been the major factor, like I initiated by significant followed by atomic damage has been the major factor, like I initiated by significant followed by atomic damage has been the major factor, like there would be factors followed by which is food and threat. It is more or less similar like there would be factors aggression initiated which is food and threat. It is more or less similar and in case of human there would be again property damage followed by which is major threat. It is more or less similar and in case of human there would be again property damage children which is major basically and in case of human there would be consider this again cats still consider children which is major humans may be basically and in case of human there would be amusement again cats still consider children which is major. Now coming to the main part humans may be basically and in case of human so amusement again 10 years back in 2000 now coming to the main part damage basically and in case of in her study that again 10 years sites human now coming to the main part damage basically and in case of in her study it's the same case 10 years sites human now coming to the main part damage basically and in case of in her study so it's the same case 10 years sites human now coming to the main part damage basically and in addition in her study so it's in every case 10 years sites human now coming to the main part basically and in addition in her study so it's in every case 10 years sites human now coming to the main part basically and in addition in her study so it's in every case 10 years sites human now coming to the main part basically and in addition in her study so it's in every case 10 years sites human now coming to the main part basically and in addition in her study so it's in every case 10 years sites human now coming to the main part basically and in addition in her study so it's in every case 10 years sites human now coming to the main part basically in a trail, I've considered fear and attacks. There are two categories. Attacks when there is an actual attack happening, and fear when only the human person. Fear and property means like anything. Like if the macaque is on attacks when there is an actual attack happening, and like especially in residential areas, they tend to be on these property structures means like anything. Like if the macaque is on even if they are not per se damaging anything, a lot of times people do chase them away because they expect damage. Even if they are not, those kind of aggressions are considered damaging. A lot of times people do chase them away because they expect damage. Even if they are not, those kind of aggressions are considered damaging. A lot of times people do okay. So general perception is quite interesting because I would like to tell one particular incident where I met one person in two different sites and his thoughts on macaques were completely different. I met this police officer in Dajkali temple and there he had he was really like he was not against macaques. He was like okay, they are with the goddess. We have to protect them. We have to feed them. It's a blessing for us that we get to do this. But then, coincidentally, I met him in Buddha temple. His house was there, and the macaques were running around on his roof, and he really wanted me to capture them and take them away. His house was there because he thought I was were running around on his roof, and he really. I feel it depends from side to side. Like in Buddha temple, he had his house, so he was worried about that more. And in Kali temple, he didn't really have to worry. It depends from that side to side. So, like, yeah, even for one person, I think the perception changes worried about that more. And in Kali temple, he didn't really have to worry. It depends from that side to side. So, like, yeah, even for one person, I think the perception changes worried about that more. And in Kali temple, he didn't really have to worry. It depends from that side to side. So, like, yeah, even for one person, I think the perception changes worried about that more. And in Kali temple, he didn't really have to worry. It depends from that. I had a slide which showed intergroup aggression, and this was only shown in Lachiwala and Kali temple, and the incidences were very less. I had a slide which showed intergroup aggression, and this was only shown in Lachiwala and Kali temple, and the incidences were very less. I had a slide which showed intergroup aggression, and this was only shown in Lachiwala and Kali temple. No, but I know that across the sites, more or less the number of troops are similar, like with overlapping home ranges. In campus, there are three. In Lachiwala and Buddha temple and Kali temple, there are two with overlapping home ranges. I don't think that will like make any Lachiwala and Buddha temple and Kali temple, there are two with overlapping home ranges. I don't think that will like make any Lachiwala and Buddha temple and Kali temple, there are two with overlapping home ranges. I don't think that will like make any Lachiwala and Buddha temple and Kali temple, there are two with overlapping home ranges. I don't think that will like yeah, Lachiwala and Buddha temple and Kali temple, there are like the frequency did increase in March and April. I don't think that will yeah, Lachiwala and Buddha temple, especially when newborns are there, like the frequency did increase in March and April. I don't think that will yeah, Lachiwala and Buddha temple, yeah, I just mentioned there, like the frequency did increase in March and April. I don't think that will yeah, Lachiwala and Buddha temple, yeah, I just mentioned there, like the frequency did increase in March and April. I don't think that will yeah, Lachiwala and Buddha temple, Yes, actually, the truth movement does go outside the campus territory, and most of the dairy produce incidents have been seen there only, and it does go outside the campus territory, and most of the dairy produce incidents have been seen there only, and it does go outside the campus territory, and most of the dairy produce incidents have been seen there only, and it does go outside the yeah, please. And most of the dairy produce incidents have been seen there only, and it does go outside the yeah, please. I'm not really sure, but I think even if food availability, food is not scarce, even then macaques will come. I think even if food availability, I believe food is not there are other, even then macaques, sorry, I'm wrong. I think even if food availability, I believe food is not there are other, even then macaques, sorry, I'm wrong. I think even if food availability, I believe food is not there are other, even then macaques, sorry, I'm wrong. I think even if food availability, I believe food is there are other, even then macaques, sorry, okay, so with respect to the sticks, it will work, but for some time, because I've already seen macaques exploring the sticks and the basket. So, I don't think it's a permanent solution, because I've already seen from my study what I've realized and the basket. If humans are a little more, I don't think it's a permanent solution, because I've already seen from my study what I've realized and the basket. Okay. Humans are a little yes, I don't think it's a permanent solution, because I've already seen from my study what I've realized and the basket. Okay. Humans are a little yes, I don't think it's a permanent solution, because I've already seen from my study what I've realized and the basket. Okay. Humans are a little yes, I don't think it's a permanent solution, because I've already seen from my study what I've realized and the basket. Okay. Yeah. I don't think it's a permanent solution, because I've already seen from my study what I've realized and the basket. Yeah. I don't think it's a permanent solution, because I've already seen from my study what I've realized and the yes. Yeah. I don't think it's a permanent solution, because I've already yes, sir. Thank you. Yes, sir. Thank you. Yes, sir.
Right, understanding the impacts of anthropogenic changes on soil distribution and habitat association. Mountains as an ecosystem is something that wants to explore in the future. David, the field is yours and all that.
Good morning, everyone. I'm David. I'll be presenting my dissertation titled Leveraging Citizen Science for Bird Monitoring, a case study assessing the impacts of urbanization on bird assemblages in the Nilgiris. So, what does urbanization do? Urbanization is one of the most adverse forms of habitat modification, which leads to structural homogenization. Most cases, buildings, roads, parks, etc., that we see in all neighborhoods, which creates a set of ecological filters that filters the regional or local species pool and favors a few select species, thus resulting in numerous losers and a few winners, thus leading to biotic homogenization in the community. Citizen science as a tool for data collection is well used in the Western world, where the interest in birding and volunteer support are also high. They are also logistically very helpful in collecting large scale data, as it is less labor and time intensive for a small group of researchers to do compared to a small researchers group to do. But it's also important to know that there's a lot of biases in doing by following this method. The major ones are clustering of data, and the effort is not uniform, and the observer quality is questioned. So these biases are predominantly present in unsystematically collected citizen science data, which are examples like eBird, um, biodiversity portals, etc. But in case of systematically collected citizen science data, where a studied, well implemented study design takes care of clustering of data and effort, but still observer bias might be present. But in case of researcher grade data where observers are trained, these uh, biases might not be present. So in order to answer, in order to use citizen science to, my objectives are to use systematically designed citizen science surveys to understand the effects of urbanization on the bird community parameters in the Nilgiris, and to compare systematically collected citizen science data set against a researcher grade data set. My research question follows the same, which wants to look at drivers, effects and potential drivers of urbanization um, on species richness, abundance, and composition of bird assemblages, and also occupancy of select species. And also to see if there is a observer bias in the citizen science data. So my study area is comprised of three cities: Ooty, Kunur, and Kotagiri, present in the elevated Nilgiri plateau. Together, combined together, they cover a approximate area of 100 square kilometer. The extent of each city was mapped using a combination of nightlight data and a land use land cover map. So I adopted a bird atlas framework for study design, which basically divides the whole study area into 1 square kilometer grids, which are further divided into 4 quadrants, each 500 into 500 meter grid cells. Uh, for one 500 to 500 meter grid cell was randomly selected per 1 square kilometer grid, and a 300 meter trail was laid in each 500-meter grid cell, which was also made sure that it cuts across the majority of the habitat types present within the grid. And these transects were also time bound, 15 minutes. So also classified the grids based on the percentage of urban cover. So all grids which had more than 30% urban cover were classified into class 1, 30 to 50% urban cover into class 2, 15 to 5% into class 3, and less than 5% into class 4. For citizen science surveys, teams of 2 to 3 members comprising a expert birder and amateur birders were constituted, and they surveyed all 97 of my grids, and each grid was replicated 3 times temporally. For research grade surveys, I sampled 25 grids, random grids out of the 97 grids, and I collected data along the same trails used by the volunteers to keep the effort constant. And each grid was replicated 3 times temporally. These are my habitat covariates, where I calculated percentages of each land use land cover class, land use land cover class within each grid cell, and distance to Shola from each centroid of the grid to the nearest Shola patch, and distance to city center from each of the grid cells. I also asked the volunteers to collect vegetation structure data, which is canopy cover and understory cover, which were ocular estimations um, between low, medium, and high. And the land use land cover map was borrowed from Ramesh et al. 2022. So moving on to the results, richness in- decreased with increase in urbanization, and the highest levels of the class 1 had the lowest, and the class 4 had the highest. Abundance showed an opposite trend where uh, with increase in urbanization, there was an increase in abundance. So the highest urban class had the highest abundance as well. Rank abundance curve indicates that at higher levels of urbanization, the community is more uneven, and at lower levels of urbanization, the community is comparatively uh, more uneven and more even. And in higher cases of urbanization, it's more uneven. To compare the composition between the urban classes, I used NMDS, which indicates that the bird communities between the four urban classes were distinctly different, which was also supported by the Parmanova results. To look for drivers that explain the trend in richness and abundance, I performed generalized linear models. Uh, First, a global model including all the covariates was created, and the dredging of the global model resulted in the best model with the lowest AIC. So for richness, four predictors explain the uh, most amount of data, out of which distance to shola and percentage of settlements were significant. In case of abundance, the same four variables explain the majority of the data, but in addition to uh, percentage of settlements, distance to city center and shola forest were also significant. So these are response curves from the best models for species richness, where you can see on the left, with increase in settlement, there's a stark decrease in richness, and although distance to shola has a very weak relationship, but still there's a decrease in richness with increase in distance to shola. For abundance, on the top left, with increase in percentage of settlements within each grid, the abundance increased, and with increase in distance to city center, the abundance decreased. And the same trend was seen with the percentage of shola cover within each grid. So I classified species based on their ecological trait, and forest specialist species such as grey jungle fowl, black and orange flycatcher, sholakili, laughing thrush showed a decrease in occupancy probabilities when there was an increase in urbanization. Um, Urban generalist species such as house crow, rock pigeon, house sparrow, jungle myna showed the opposite trend where they increased with the occupancy of their occupancy is increased with increase in urbanization. Whereas for open area generalist, they didn't increase or decrease, and they remained more or less constant across the four urban classes. So in order to compare between research grade dataset and citizen science dataset, RG and CS, I calculated encounter rates for species between the two datasets. And although there were minimal differences in species between the two datasets, none of them were significant except for the Indian white-eye, which I have highlighted over the red box. So small and flashy species on the left were more or less detected equally between the two datasets, and there was a slight difference in small and shy species. Um, but in case of large-bodied species, whether they are forest or commensals, they were all detected more or less equally by the two datasets, and none of them were statistically significant. I also computed community dissimilarities between the two data sets as data was collected in the same sites between the two data sets. So um, most of the grid sites had a very low dissimilarity in the community that they captured. So key results and conclusion: urbanization reduced species richness and increased abundance. Percentage of settlements, presence and distance to shola patch drives the observed patterns in species richness and abundance. Forest specialist species were replaced by commensals, and generalists were not affected by the urbanization gradient. And also found that systematic citizen science data is comparable in accuracy to research grade data. I would like to acknowledge and thank the following people for their support and guidance throughout the course. Thank you.
If you model out instead of occupancy, abundance, what is it?
More or less the same.
Better response because your
Right, sir.
confidence intervals are very large.
Very large.
That will improve or not?
It will improve.
One more thing, do you think the life street rates are a big filter between forest to city gradient?
I think so, sir, because in this case, my forest specialist species are primarily understory insectivores, and they are also endemic to the narrow endemics to the Nilgiri Plateau. So I think stark difference in the forest and understory cover within these grids are a big limiting factor for their presence and movement. So I think that becomes a big filter.
Maybe we didn't say, but I was about to ask the same question. Were you expecting house style of the forest and then you contributed to urbanization? It's a evolutionary thing. So most of your species which are shown in urban areas is because of their evolutionary traits, they do not exist in the forest. It's all that evolution is why it's house style, why not jungle style? So it is that significant. So if you would have put that evolutionary aspect into your analysis, then you probably have been able to tell us, okay, these are the species which are evolved because of their presence in the urban areas, but these are the few species which are differentiating because of the rural urban.
Right.
Is if we convert Delhi today into a forest, a lot of Himalayan species will be there. We convert it back, we will lose some species, but we will have some other. So the difference between what actually are the species which we lose because of this. So that if you just I need the naturalistic takes even that of concept.
Sure, sir. I'll switch it with.
Give you a better picture which actually what are actually users of this. And as sorry, sorry. So you also talked about that you verified this by the researcher-based sampling. And you're not sure the results of that. Have you?
Sir, I compared encounter rates between the two data sets.
What was the time frame?
So all were collected within a 5-day period, both the data sets. No, so major, so citizens did all 97 grids. So that was across 3 months time frame. So whenever they are doing this, the 25 selected grids that I want to do, I will also do when they are also doing the same time. So it was within the same time frame, let's say within a week. So I assume the community to be closed.
So do you visualize that in future you will use citizens and
Right. It should be a combination. Right. That I was planning to do, but then inherently all these cities in the Nilgiris are quite wooded cities. If you have been there, they are very green cities. So there is not really a grey and a green space within the city. Everywhere there is uh, wood gardens within houses. So hmm.
Online doesn't tell us. But this change by Rohit here, she says, looking at two birds, not literally, birds not here, studying bird assimilators and assessing human things. Very nice study. Question is, what might be the reason for difference in encounter rate in data sets for wildlife? India wildlife.
Oh, right. So I think white eyes are also very drab looking birds in the Nilgiris at least. Um, they are gregarious, but I think I don't really can explain it quite well, except for there's a bias in small drab looking birds. And then just because there was a stark difference, the test picked out it as a significant. But then even for Nilgiri flycatcher, the medians are quite different, and there's a different. So I think overall birders in general are having a difficult time identifying this in the landscape. Any of the four species on the
Moving on to the next video, I call upon Namita Madhu, who will be talking on the impact of local and landscape level assets on rural communities. Namita here is another budding community ecologist with interest in forest dynamics, agroecosystems, and the impact of forest. All of us, Namita, you can take over.
Thanks, Manas. A very good morning to everyone present here. So this project deals with the effects of local and landscape level drivers in influencing bird diversity and persistence in cashew plantations in the northern western Ghats. So agroforestries have been expanding across the globe with crops like tea, rubber, cashew and coffee prevailing in our country. They have been generally promoted as biodiversity friendly, especially when compared to other types of agroecosystems. However, the general trend is that agroforestry negatively impacts bird species richness. Different forms of agroforestry differ in the magnitude and direction of their impacts. For example, in this case, we have oil palm that have severely negative effects on species richness, at least as compared to the other crops like coffee and cocoa. And these impacts are particularly high in the Oriental realm to where we belong, to which we belong. Let me take the example of the Western Ghats, where we see a major change in land use. So you see these light blue patches in the figure, right, in the map. These light blue patches represent plantations, which are especially concentrated in the southern region. In the southern and central regions of the Western Ghats, we have tea and coffee that have been given more attention with respect to impacts on biodiversity. But in the northern regions, we have crops like mango and cashew, which have been relatively poorly studied. Cashew has been expanding at an alarming rate in the northern Western Ghats. In fact, in Sindhudurg district, in which my study area is located, it contributes to about one-third of Maharashtra's cashew cultivated land. And in this map, you see two tehsils of Sindhudurg, Savantwadi and Dodamarg. And in this map as well, about one-third of the landscape is dominated by cashew monocultures. And a large proportion of land that's been given or that's been provided for these plantations comes from forest, especially private forest, which is located at the lower elevations. These forests have been provided less conservation attention, at least when compared to the higher elevation forests.
When we look at the impacts of agroforestry and biodiversity, we look at it through two lenses. So one is the site level factor, and native trees contribute to this because they contribute to structural complexity and diversity of vegetation. And quite often the plantations that have native trees within them follow less intensive management. But we also look at the lens of landscape, at the landscape context. So in this case, in my study area, I have an agroforestry matrix where we have a plantation landscape with forest patches interspersed in between. So the role of forest patches is quite huge in biodiversity maintenance, especially in agroforestry matrices. When we look at these responses on birds especially, we look at it through, again, different perspectives. The first is taxonomic diversity, where we treat all species as equal. Then there's functional diversity, where we have different functional roles that are played by these species. In this case, we have nectarivores, invertivores, granivores, and frugivores. And in phylogenetic diversity, we look at the evolutionary relationships between species. So the more phylogenetically diverse a community is, the more species that it has that are evolutionarily different from each other. These metrics, especially community metrics such as functional and phylogenetic diversities, give us information on community assembly processes. For example, we have a clustered community here in the sense we have species that are similar to each other functionally as well as phylogenetically. Here we have two bulbuls, two plenials, and one tailor bird. Functionally, we just have frugivores and invertivores. But when we look at an over-dispersed community, we see that these species are quite different from each other in terms of both evolutionary relationships as well as functional roles in the ecosystem. So we usually infer clustering as an impact of environmental filtering, and we usually look at over-dispersion as an impact of competition. Similar species cannot possibly coexist. So we have species like the Malabar grey hornbill and the little spider hunter that may contribute to phylogenetic diversity, resulting in over-dispersion. But not only are community metrics important, species-specific information is also very important because we have range-restricted, threatened species and forest specialists that provide interesting responses to land use change. And when we have community responses that differ, for example, if we see a signal in functional diversity but not in phylogenetic, then the mechanisms behind that could possibly be explained by species-specific responses. Given this background, I have two objectives. One is to look at composition and taxonomic, functional, and phylogenetic diversities, how they differ between these land use types of forest, mixed cashew plantations and pure cashew plantations. And the second is to look at species and trait-specific responses to local and landscape level factors, and do phylogenetic relationships influence these responses. So my study area is in Tilari, which is in Dehradun, southern Maharashtra. This is situated in the northern western Ghats. This is situated in an agroforestry matrix where you have cashew plantations and forests. These cashew plantations are varying in proportions of cashew trees and native trees. So the yellow triangles in this map represent mixed cashew plantations. The red ones represent pure cashew plantations and green ones represent forest. The range limits of several species end here, like for example, we have Draco and grey-headed bulbul, Malabar woodstrike, Microxylus uttaraghati, and the hump-nosed pit viper. This is how I sampled during the study. I set three land use categories. So I talked about forest, mixed cashew, and pure cashew land use types. I'd like to define mixed cashew plantations before moving further. Mixed cashew plantations are those plantations that have varying proportions of native trees interspersed in between. The minimums, so we set 100 points, 25 each in forest and mixed cashew and 50 in pure cashew, and sampled between January to April. The minimum spacing between points was 250 meters. These were the parameters that we recorded for vegetation sampling in 10-meter radius circular plots. I sampled birds along with my field collaborator Vishal in 10-minute point counts, replicated 5 times. So each point was replicated 5 times. These were the parameters that we recorded, and we sampled in the mornings from 6:45 to 10. So this is how I analyze my data. For characterization of land use types, I used vegetation information. I used hill channel diversity for tree species diversity, log transform the structural attributes, and then use linear models to test for the effects of land use type on vegetation attributes. I followed this by ANOVAs and TIMKIs for pairwise contrasts. For landscape, the characterization of land use type is used for local level information. For landscape level information, I used forest cover and cashew cover. I obtained the optimum buffer radius using the scale of effect approach outlined by Farid. So the relationship between species richness of a point with the amount of surrounding forest cover peaked at 100, 200 and 800 meters, after which I chose 800 meters as the landscape variable and obtained forest and cashew covers for that radius. For the first objective, which compares, composition, taxonomic, functional, and phylogenetic diversities between these land use types, I obtained trait information from AVNet and Elton traits to online databases which have morphology, diet, and other kinds of information on birds of the world. And I obtained diet data from Ali and Ripley's Handbook of the Birds of the Indian Subcontinent. I obtained phylogenetic trees from birdtree.org and pooled temporal replicates for these analyses. So for species composition, I used NMDS, an ordination method with Gray-Curtis dissimilarity index, followed by Anosum and Permanova. For taxonomic diversity, I used Hill-Shannon, which is basically a sample coverage-based rarefaction method, which accounts for sample coverage. For functional and phylogenetic metrics, I used the standardized effect sizes of the mean pairwise distance. So, mean pairwise distance can be defined as the average functional or phylogenetic distance between all possible species pairs in a community. So, I computed plot-wise SES values of FMPD and PMPD, square root transformed them, and followed it up with LNs for the effects of land use type on these metrics. Again followed by pairwise contrast analyses. So this is how the result would look. For the second objective, which looks at species and trait-specific responses to local and landscape level factors and if phylogenetic affinities influence these responses, I used HMSC, which is Hierarchical Modeling of Species Communities, a joint species distribution modeling framework to assess three things. One is trait responses to local and landscape predictors, the second is species responses to local and landscape predictors, and third is a phylogenetic signal in the residuals to test for the effect of affinities. Rare species that occurred at less than 5% of sampling points were excluded. Pooled temporal replicates were used for this analysis, and the spatial random effect was set at the level of the point location. I used a presence-absence model with Probit link to model occurrence probabilities of each of the 70 species across land use types. So these were my predictors. For local scale, I had land use type, which was set at the level of a patch, which differed in radius from 100 to 400 meters. Then I had two landscape level variables, forest cover within 800 meters buffer and cashew cover within 800 meters buffer. I obtained the land cover data from the M.Sc raster by Anushka Reddy. And like I mentioned before, I used the scale of effect approach for getting the optimal buffer radius. This is the statistical framework. So HMSC basically gets the results of occurrence probabilities using four things. One is environment data, phylogeny data and traits data which are incorporated into the species, And all of this is set within a spatio-temporal context. So these are my results. So for trees, I recorded 56 species belonging to 29 families, and I recorded 102 bird species belonging to 38 families. Forests have generally better vegetation structure. They have higher tree species diversity, tree height, and canopy cover than both cashew habitats. These were the functional traits that I selected to account for trophic, locomotory, and diet traits. Then I also included two other variables, migratory status and habitat preference. Distinct communities were recorded across land use types. These three were distinct, communities. Pure cashew had least taxonomic diversity, followed by forest and then mixed cashew, but the CIs of forest and mixed cashew overlapped. When it comes to phylogenetic diversity, pure cashew had the least, and it had significantly lower SC, ST, MPT than the other two land use types. But the interesting thing is that functional diversity did not differ across land use types. The HMSC revealed that the site level factor had greater explanatory power. So basically, the land use type representing vegetation attributes had the highest explained variation, 58%, and the landscape variables, which explained 15% for pure cashew and 12% for, not pure cashew, for forest, for cashew cover in 800 meters and 12% for forest cover in 800 meters. The evergreen trait and the sedentary trait responded significantly to predictors. So evergreen species responded negatively to land use change, and sedentary species responded positively to the landscape variable of forest cover and 800 meters radius. So when we look at species level responses, I was looking at forest specialists and open species. So forest specialists were found to lose out in cashew because they lost their habitats, right? So this is how it's generally modeled. So occurrence probability is modeled across these three land use types. So we have a total of 18 species that responded negatively to land use change. This similarly for open species, we have the occurrence probability on the y-axis and the land use types on the x-axis. And we have 19 species that responded positively to land use change. So they went to cashew, they went to land use change. I also found that there is no phylogenetic signal in species responses. That means that there is no evidence of niche conservatism. Niche conservatism can be defined as traits that are, the phylogeny is conserved within traits. So when I say that there's no niche conservatism, I mean that species that are evolutionarily similar to each other did not necessarily have similar ecological niches. So what do these results mean for the conservation of birds in this landscape? So I found mixed cashew to have an intermediate level of disturbance and hence have elements of both forest and open habitats. I found that there was no signal in functional diversity, right? So functional diversity did not differ across land use types. So this may be because of ecological replacements occurring at the species scale. For example, we have the yellow-throated bulbul, which is a frugivore, replaced by the red-belted bulbul, which is again a frugivore. Then we have the greenish gobbler, an invertivore, replaced by the common tailor bird, an invertivore. So the functional roles are maintained, but the phylogenetic relationships are lost. So there is a loss of phylogenetically distinct lineages, which is reflected in phylogenetic diversity when land use change happens. So although some forest specialists can be supported by mixed cashew plantations, for example, we have the small minivid, the jungle owlet, and the Malabar whistling thrush that are supported by mixed cashew plantations. Some are being filtered out in both types of cashew habitats. So we have the black-necked monarch, the white-rumped shamba, the emerald duck, and the Malabar trogon that are being filtered out and replaced by these open species like the red-whiskered bulbul, the Premier, Ashigrenia, spotted dove, and peafowl. And These results suggest that mixed cashew is a relatively less intensive agroforestry, at least when compared to pure cashew habitats. But forests are irreplaceable for many forest species like the Malabar turgon over here. So the thing is, my HMSC results suggested that the local scale variables were more important. However, there were some species that responded significantly to these landscape variables. So we have four species responding negatively to cashew cover within 800 meters, two of them responding positively to it, and then three species like the grey-bellied cuckoo and these two warblers that respond negatively to forest cover within 800 meters. But the thing is, these results would not be obvious if not for species responses. So hence, it is important to look at species-level responses as well. So these results suggest that we could conserve birds by making cashew plantations more biodiversity friendly, but as well as retaining forest patches in the landscape. So these are my references, and these are the people that I want to thank. So thank you. Thank you. Yes.
The function that I'm audible.
Yeah.
So the function diversity results, were always really putting the marker space to one direction for all your languages? Because they're generally big outliers on your marker space.
Absolutely, I agree. They are very original and they have a high morphological originality. Um, the thing is, I just had one species that responded significantly to these predictors. But so, which was the Malabar grey hornbill. So it might not have pulled it that much. It might not have influenced the data that much. But
Maybe a good approach would be to just run one more set without the marker, just to see whether those replacements are happening for other species as well.
absolutely, I
It gets skewed because of the marker.
yeah, it's possible. Yes.
Otherwise, it will be.
Thank you.
I have just one question suggested about keeping a mixed cashew plantation.
Yes.
Any idea by losing one cashew tree what loss it is? Just a broader idea.
By losing one cashew tree, uh, losing one in the sense, are we replacing it with a native tree or right, right? So when this happens, right, when we have mixed cashew plantations, they have trees in varying heights and they have trees that provide sources of fruits and nectar to a lot of birds, which I observed.
Not that
Oh, I'm sorry.
you suggested that move towards mixed cashew,
Yeah, right.
so people will get lost. So it's nice to suggest, but what is that level of loss which they incur, whether they will agree to that kind of.
Right, so understandably, yeah, I definitely get this. So, the thing is, there have been no studies that look at the economic impacts of mixed cashew plantations as compared to pure cashew, which I think should be done because when I went to sample, I observed a good yield of fruits. But the thing is, the fruiting happened later when compared to pure cashew plantations. So if the fruiting is a little later, but it's just mixed with other native trees, it may not necessarily have a negative impact on uh, economics. But the thing is, I can't confirm this without evidence. So this is just based on my field observations. But from what I observed, they have a relatively good yield.
And one more question about that. Most of your forest and cashew are still embedded in natural forest surrounding.
Yes, I agree, absolutely.
That do provide a kind of a community which otherwise if you have a cashew plantation far away may not necessarily be. So it's a context uh, interpretation because that matrix, right?
Right. Uh, I'm
Yes,
sorry,
what
I
I mean
didn't
is,
understand.
as the distance from the forest patch will grow,
Yes.
beyond a point, it will have a drastic effect on your community.
Beyond a point. Um, so I decided the buffer radius based on that approach, right? So it was just to look at how one metric is maximum at one certain distance. So I chose 800 meters because it peaked. The species richness at a point peaked versus the amount of forest cover in the circular patch of that radius. So I feel that effects may not be as significant as it is at 800, beyond 800 meters. That's what the rationale of this was.
Do you have any online questions? There's one question from Rohit. Interestingly, here since point level features of factors seem to be more important than heterogeneous landscape, like the study area, what policy instruments are available to encourage these cash economies? Should governments push economic incentives like payment for ecosystem services and framework where to retain native trees on site?
That's a great question, Rohit. Thank you. um, so there are, I think there could be implications for certification, but this cannot be, you know, this cannot be said for sure without surveys and interviews with farmers and cashew plantation owners. What I so was Rohit asking for conservation? I mean for policy recommendations? Right, right. There's already one rule, the Maharashtra Tree Felling Act, which basically bans the felling of certain native tree species. However, this is not checked. This is not regularly checked at our in the landscape. So I recommend, and what I would recommend is that inspections be done to check for the compliance of this act, and also just interviews with farmers, a lot of interviews with plantation owners to see what they think and to see if they would be motivated by mixed cashew plantations. Yes, that would be nice.
Do any birds cause damage to cashew? Anyone?
Damaged
Damage
by.
to cashew. Do they eat cashew or damage?
Oh, right. No, not necessarily. No, none of the bird species, at least based on my observation, none of the bird species damaged.
Not even to the fleshy food?
Right. So, yeah, there were some frugivorous interactions, but again, since the seed itself is toxic, right? They don't play a role in dispersing it, but they, yeah, like you said, they just feed on
No
the fruit.
economic loss.
No economic loss because it's very negligent. Birds also, for some reason, don't like cashew that much. Only a few interactions were observed on fruit.
This from Dr. Salvana. Great study. What is the variability amongst your sites in terms of intensity of plantation and forest cover in a region?
Can you please repeat, Akalan?
What is the variability amongst your sites in terms of intensity of plantations and forest cover in a region?
Okay, across. Okay, okay. I mean, there was a gradient in land use type, which I recorded in this study, right? But other than that, there's also a gradient in the use of the type of cashew. For example, most, if not all, actually almost all mixed cashew plantations use the local native variety of cashew, which is called gouti in the local language. They use gouti cashews, which are bigger, The branching was more dispersed, they had a wider level of branching, and the trees were generally a bit taller. But in pure cashew plantations, the trees were generally small, which were of Bengulla variety, right, 1 to 7, which are commercially important for the people in the landscape. So they were shorter, they were more homogeneous, and they relatively had a greater spacing between each other. And there's a gradient in the use of pesticides. Pure cashew plantations use pesticides, and mixed cashew plantations didn't use pesticides. And pure cashew plantations also use pesticides, which were again not used in mixed cashew plantations. Then, in terms of forest cover, of course, I could not disentangle the effects of secondary forest and primary forest. That information was not available, unfortunately. So I just obtained the amounts of forest cover around 800-meter radius for primary, I mean for any forest cover in general. So yes, I observed that gradient as well, which I recorded in the study. So the thing is, I set 50 points in pure cashew because I wanted a gradient of forest cover. So I set 25 points near forest, 25 cashew points near forest, and 25 cashew points away from forest. Hence, it will account for the proportion of forest cover around the point. That was the rationale behind that aspect of the study.
Thank you for the questions.
Thank you, Nanda. Thanks.
So for the next presentation, I call upon Niyaz Ahmed, who is going to talk about health risk assessment of the habitat and diet of smooth-coated otters. Niyaz here is interested in otter ecology, and this study aims to answer how pollution is impacting otters. All the best, Niyaz. Over to you.
Thank you, Manas. So, good morning to one and all. Myself, Niaz here, and today I am here to present and defend my master dissertation project. As we all know, we, the human beings, epicenter of destruction to the entire environment as ecosystems. We are destroying this environment throughout many centuries. But according to the Living Planet report, there has been significant decline in the past 50 years with respect to the wildlife populations, and the major habitat which has been destroyed with respect to these within these 50 years is freshwater habitats, with a decline of 83% in wildlife population. And the major threats which are considering causing the loss of these freshwater habitats are grouped into 6 categories as overexploitation, flow modification, habitat degradation, climate change, invasion by exotic species, and water pollution. Water pollution is also a major account of all these above mentioned 5 categories. The water pollution is categorized into 2 sub-categories such as non-persistent pollutants and persistent pollutants. The non-persistent pollutants are not a very good are not a very great threat to the freshwater habitat, but whereas the freshwater pollutants such as but whereas the persistent pollutants such as plastic and heavy metals which remain in the environment for a very long time by entering and traveling to various food chains and affecting the ecosystem as well as the habitat. To, uh, to assess how persistent pollutants affect, we cannot look into each and every aspect of a food web, but we have to see the top apex predator which is present in the habitat. So smooth-coated otters or otter species are the top apex predators of freshwater habitats in Indian rivers. They are also the better biomonitors than bioindicators because whatever the heavy metals or persistent pollutants present in the environment are definitely going to bioaccumulate in the top apex predator. They are also called as sentinel species. It is nothing but the species which are seen or experimented to see the negative effects which might be causing to the humans. Now coming to the based on now based on the persistent pollutants and otters as apex predators, there are about 53 research questions have been answered in the entire world. And with respect to Indian questions, only 2 research have been done. That is one which was originated in WII, that is Dehradun, other is in Goa University. Now there is a very large gap because India is a developing country. There is a need for such studies, so I entitled my topic for my dissertation as risk assessment of potentially toxic elements in the habitat and diet of smooth-coated otter in Tungabhadra Otter Conservation Reserve, Karnataka. Now let's understand what is this Tungabhadra Otter Conservation Reserve. It is the only conservation reserve for the protection of otters in the entire India. There are about 125 mines in and around the 60-kilometer radius of Tungabhadra Otter Conservation Reserve, the, which are directly connected through mining streams to the study area, that is conservation reserve. And the first mining here was started in 1907, which indicates a large amount of mining has been taken place in this area. The major type of mines here are of iron and manganese. Now, with respect to this, the first objective I want to discuss here is how the otters are distributed in this landscape and what type of specific distribution pattern they follow. To test this, the field method that I implemented was science survey by dividing the entire Tungabhadra by dividing the entire TOCR into 500 meter and 500 meter grids. I recorded the science that was I recorded the size. A total effort of 100 kilometer was intensively surveyed by foot and board. And the different types of signs that were encountered are as follows: direct sightings, grooming sites, otter tracks, pug marks, sprain sites, and also the leftover prey. With respect to this, the the entire TOCR, which was divided into grids, for each grid a sign density was calculated. For the total number of grids was 112. The sign density was calculated with respect to the number of signs that I encountered per grid divided by total distance that I have walked in the grid. That accounts to the effort. Now the entire 112 grids were equally divided into three categories as moderate, low and high density grids. Later, Moran size spatial autocorrelation was used to test the distribution pattern. Now coming to the results, a total of 132 signs were recorded in the study, and the major signs were of sprains as well as sprinting sites and the least signs of pugmarks. Now to see the distribution pattern, when all this data was incorporated into GIS platform, we can see some clumps of distribution. But when Moran site test was ran and we found the value to be plus 0.99, that is very close to plus 1, it indicates there is high spatial autocorrelation of these signs, which indirectly indicates the clumped distribution of otters in this landscape. Now coming to the second objective, I wanted to see what is the dietary spectrum of otter in this human-dominated riverscape. Why human-dominated? Because of the inclusion of fishes by the National Fisheries Board. So for this, firstly I prepared reference slides by collecting the fishes from the fishermen and removing the scales. And each scale was characterized with various characters to identify the species level, such as the anterior, posterior, and various characters. Then later, the dyes called as alizarin red dyes and DPX mounting agent were used to prepare permanent reference slides, which will be later used to analyze the sprains, to analyze the scales which are present in the sprains. Now, coming to the methods that I use to analyze the sprains, there are mainly two methods. That is frequency of occurrence method and score bulk estimate method. The frequency of occurrence method is just the relative frequency of a category present in a sprain. But whereas score bulk method is a revised version in which for entire sprain we will be visualizing the score from 0 to 10 with respect to the species of scale present in the sprain. So firstly, so firstly we sorry. So firstly we analyze the major prey categories present in the sprain, such as fishes, crabs, bird, etc. Next, a subsampling method was was implemented to estimate the number of fish species present in the sprain. For which, first, if we score entire scat as total score value of 10, then randomly we will be selecting 10 scales. If all the 10 scales are belonging to the single species, then we give the entire sprain as a score of 10 to the species A. If we get any new scales of any other species, when we are first randomly selecting 10 scales, then we again select next set of 10 scales, and this process continues till we estimate all the species of fishes in the sprain and give the score with respect to this. Now coming to the results, about 97% of diet of otters in Tungabhadra were consisting of fishes and 2.13% were of crabs. And with respect to species level, there are 15 species belonging to 6 families, but one species was unknown. Because I was unable to prepare the reference slides, so I couldn't identify the particular fish. But about 4 species of fishes were non-native and 10 were native, which and about 70% of diet of otters in the landscape was of non-native species, and about just 30% was about native fish species. Now coming to the third objective, as in the first objective I divided the density of waters respect to low, moderate and high density. So I wanted to see the correlation, so I wanted to see this in relation with respect to heavy metal concentration, for which firstly I collected samples from study area that is TOCR and control site, which was selected on the basis of divide of any mines in and around it, and we collected water and sediment samples. The water and sediment samples in TOCR as well as control site were selected from three parts of a site, that is right bank of the river, left bank of the river and the center of the river, and they were mixed up. About 200 grams of sediment and half liter of water was collected. The water was preserved using 2% nitric acid. Now coming to the fishes, the fish samples were collected with the help of fishermen such that we instructed them to collect the fishes without any metal contamination, which would differ our results further. Later, we collected the scat samples during the science survey itself. Now, for the sample preparation and analysis for analysis of elements present in these samples, there were majorly three steps followed. That is homogenization. It means that mixing of all the samples within it. For example, water sample was homogenized using centrifuge. Sediments and scat samples were homogenized differently using granite mortar and pestle, and whereas fish sample were homogenized using REM homogenization. The next step comes is digestion. Digestion is a process that is done to remove all the biodegradable material present in the sample, for which two methods were implemented, in which first was overnight digestion was done by using 5 ml nitric acid and 2 ml hydrogen peroxide, followed by microwave digestion, in which the samples were ran into different temperatures for different prolonged times to digest all the biodegradable material present in them. Later, all these samples were ran into ICP-MS to obtain the elements present in the samples. Now, later the risk assessment was undertaken using reference dose commonly used in human health risk assessment. The reference dose was used to derive the maximum allowable concentration, which represents the maximum concentration of each toxicant that can occur in prey items. The reference dose for maximum allowable concentration is calculated using the following things, that is the exposure of chemicals through prey, daily ingestion rate of the predator, body weight of the predator, and exposure frequency. Now coming to the results, in study area that is Tungabhadra Otter Conservation Reserve, in water as well as sediment samples, we found presence of almost all the metals except mercury and cadmium in water and antimony in sediment sample. But with respect to fish samples, we found presence of all the metals in all the fish samples and also in very high concentrations. And with respect to scat, we found the presence of all the heavy metals which were present in the scat of an otters. But when we did the comparison of the PTs, that is the heavy metals, between the study area and control site, we didn't find any significant difference in concentration of these metals in water samples as well as in sediment samples. But when we compare the concentration of these elements across different outer density sites, we didn't find no significant correlation between concentration and outer density sites, and also but in sediment sample we found the correlation with respect to aluminum, chromium, cobalt and nickel. It doesn't mean that wherever there are heavy concentration of metals we found high density of waters. There are various variables which might be acting on this. Now coming to risk assessment for environmental exposure for otter through water as well as sediment, we compared this with the threshold values given by US EPA, that is United States Environmental Protection Agency, and independent sediment quality guidelines. We found the PTA concentration of all the heavy metals within the threshold except aluminium in water. And the dietary exposure, when we did, we found most of the metals are causing high risk to the otters, high risk to the otters with respect to the fishes it is feeding on. Now coming to the conclusion, I want to say that there are mainly three outcomes of the study, that is otter distribution. The smooth-coated otters in Tungabhadra Otter Conservation Reserve exhibit a clump distribution pattern, and the diet composition says that otter in this landscape is feeding on 15 species, and the major proportion of its diet is composite of non-native species such as Orechomis mozambicus and Cyprinoscarpia, that is tilapia fish, invasive tilapia fish, and common carp. With respect to heavy metal exposure, we can, by the results, we are saying that all the heavy metals are below the thresholds, which may cause any effect on water with respect to water as sediment, but the otter diets but the bioaccumulation of these elements in water in further state could cause high risk to the otters in this habitat. I would like to thank all these people. Thank you.
Yes.
Yes, sir.
Found no difference in control and
Yes, sir.
experimental sense.
Yes, sir.
Do you think it is most of these metals are orogenic in nature, or there is some other thing happening in mines that you're not getting? Heavy
Sir, when we
metals.
got this result, we were also very amazed to see this. But when we again went into the literature as well as news regarding this, I came to know that the presence of Kudremukha iron ore mines, which was banned, but there was no remediation done in this landscape, which might be causing the silting of these heavy metals into these upper streams, which are considered as control sites because no presence of current mines. This could be one reason, sir.
There might be old deposition which is leaching.
Still now it is leaching, sir. Yes, ma'am. Yes, sir. Thank you, sir. Thank you, sir. Okay. Sir, during my study, I found two otters which were dead. They were dead because of various reasons. I can't confirm it. They were dead because of this presence of heavy metals. And the population estimation of this species is very difficult to say. But when there is a methodology in which we calculate the density of otters per square kilometer of length of a river, I found like 12, sir, 12 individuals. No permission, sir. The department didn't even give me the permission to collect fishes in this landscape. Those heavy metals. Yes, sir. Yes, sir. Yes, sir. Arsenic, sir. With respect to dietary exposure, arsenic is in the category of high risk. It is also with respect to toxicity reference. It's at a high risk for this anim- organism, sir. There's no, sir. There is no difference. No difference, sir. We tested for each and every element to know if there's any extra element only coming through mines. Thank you, sir.
Then I ask, lethal dose
Yes, sir.
are published for some of the metals.
Yes, sir.
So bioaccumulation lethal dose are two different. You found any difference? Because most of the metals you said are below the
Threshold
permissible
in water and sediment, sir. But when we did with respect to diet, there are like six metals are causing high risk, like arsenic, mercury, all these metals, sir.
anything above lethal dose.
Yes, sir. Mercury, as
I mean lethal dose not complete lethal dose 50 LD50 lethal dose above 50%.
yes, sir. Arsenic, this in this high risk, sir. These are above,
Above
yes, sir. Which might cause, sir, if there is more accumulation, very high risk.
one last question.
Yes, sir.
Your moral diet show cluster distribution.
Yes, sir.
Are there other environmental factors?
Definitely, sir.
What? Because I mean, on the basis of your field trip, you've not shown that. But what are the elements which are causing the concentration of otters in these sites?
Sir, the presence of big boulders and also sugarcane fields, sir. So there was previous study done in the same land case by Ankit Maur, in which he did study of habitat use, and he found that aggregation of matlab, it means that more amount of otters are present near the sugarcane fields. Okay, thank you. Yes, when we did test for the heavy metal concentration in fishes respect to native and non-native, we found more concentration of elements not on the basis of native and non-native nutrients, but the type of at which trophic level the fish are present. For example, labio species which are present, they are also omnivores which feed on algae as well as this. So they have more concentration of heavy metals. But even in non-native fish, in invasive tilapia, we also found high concentration, but not as high when compared to these other fishes. I think the ecology of fish is mainly depending on the concentration of heavy metals in this case.
Fine, thank you.
Thank you, sir.
For the next presentation, I call upon Mukul Thapar, whose study aims to understand the efficacy of investment strategies for slot better. Mukul here is another inspiring behavioral ecologist, and his study aims to improve the welfare of wild animals in captivity. Over to you, Mukul. All the best.
Good morning, all of you. I am Mukul. I'll be presenting my dissertation title as Understanding the Efficacy of Enrichment Intervention from Behavioral Responses in Captive Sloth Bears. Uh, so, as we all know that India is rich in biodiversity, which uh, usually thrive in the national parks. However, the significant portion of Indian population resides in rapidly expanding urban areas where they usually get encountered through wildlife with the help of zoos. Hence, zoo plays an important role in spreading education as well as awareness among people. Historically, there has been a significant increase in the number of sloth bears in captive facilities, primarily zoos and rescue and rehabilitation centers. This increase in the number of individual of captive sloth bears has put a concern on welfare as due to wild species, their environment changes and these kind of welfare question needs to be answered in the next studies. So the target species of my study is sloth bear, which could be identified by its crescent white patch on its chest and a shaggy coat. What makes this bear unique is its myrmephagus adaptation, lacking upper incisors, long snout, and the ability to close nostrils voluntarily. This results in diverse foraging ability to exploit food resources such as insects, seeds, and fruits, which leads them to have natural behaviors such as climbing, digging with the help of their long claws, exploratory, and foraging, etc. Black bears have been listed in Schedule 1 under Wildlife Protection Act 1972. So the objective of my study is to understand the effect of novel enrichment intervention on behavioral diversity or captive sloth bears. And the second object is being to determine the enclosure utilization which responds to the introduction of enrichment. And the third objective being the effects of zoos and enrichment on behavioral patterns, parameters such as stereotypy and inactivity. So the methodology of the study, the ethogram was modified from Fortman et al. With the help of ad libitum sampling incorporating behaviors expressed by the target population. The behavioral data was collected through scan sampling and with an interval of 8 minutes between each scans. A cumulative of 40 scans were recorded for two individuals each day. These 40 scans were divided into 4 sets consisting of 10 sets each, 10 scans each. A total of 211 hours were sampled in Delhi, whereas in Mumbai it was 257 hours. The enrichment intervention utilized for this study was pitfall feeder categorized as feeding enrichment. So the study area of my study is the first one being National Zoological Park, which is located in the heart of the country's capital, which although located in the city center spans over 172 acres. The zoo was established in the year of 1959. The Central Zoo Authority of India manages the zoo. It accommodates diverse wildlife from around the world with a focus on species which are native to India. The enclosure is categorized by open air dry moated, which spans over the area of 674 square meter. These are usually designed with a dry moat on the viewing side on of the enclosure, which acts as a barrier between the visitors as well as the animal. And it's an open air space which is covered with 3 sides with a stone wall. So the second study area is Mumbai Zoo, which is one of the oldest zoo in India, established in the year of 1861. The zoo is spread across 48 acres, housing 70 different species. BMC manages this zoo. The category of the enclosure is open air glass protected enclosure, and the area of the enclosure is 1200 square meter. So these are the comparison between the two sites. Uh, the Delhi Zoo houses 4 individuals of sloth bears. The enclosure consists of a single tree, several logs, etc., and a cave which provides shelter to the animals. It also consists of 3 shallow water troughs, which usually one is filled at all times, but the maintenance is inconsistent, which can cause concern for cleanliness and usability of the water trough. The enclosure doesn't provide any form of the cover to the animals, and the substrate is hard compact soil with patchy grass and a cemented floor at the cave site. Whereas Mumbai houses 2 individuals of sloth bears and the enclosure is soundproof. The elements of the enclosure include 5 large trees, 2 large rams, as well as 2 dens. The enclosure consists of various covers unlike Delhi. These are the covers provided by the enclosure at Mumbai Zoo: complete grass cover, tall grass covers, shrub covers, and bamboo covers. And a source of flowing water in the form of small water trough. Mumbai Zoo offers 2 substrate to the animals, one solely to increase digging behavior in the animals in the form of loose soil. One of the important difference between the site include there is no plan or schedule of enrichment activities at Delhi Zoo, whereas every weekend there is a schedule of various changing enrichment interventions provided to most animals, and hence it provides better enrichment intervention at Mumbai Zoo. This was also done before commencement of this study. So now, how can we achieve our study objectives? For behavior diversity, all behavior states and their respective frequencies were recorded to determine the frequency of occurrence of each behavior state. Also, total frequency of all behavioral states was summed to determine the proportion of each behavior state, and the Shannon diversity index was utilized to get behavior diversity. In case of enclosure utilization, according to Brereton et al. 2022, we utilized Shannon evenness index, which in our case is a measure of how evenly individuals were utilizing different zones in an enclosure. It is derived from Shannon diversity index, which quantifies the zone diversity by considering both number of zones as well as frequency of usage of these zones. And at last, effect of zoo on behavioral parameters such as inactivity and stereotypy. For this, we performed separate 5 GLMM random effect models to for each response variable as inactivity and stereotypy with two fixed effects of zoo and enrichment and a random effect of individuals. This slide shows us how enrichment affect behavioral diversity on individuals across zoo. As we can observe from the bar graph, all the individuals respond positively to the enrichment intervention, and the maximum change can be observed in Radha, one of the individual, which is the oldest individual in our sample across zoos, which could be a factor of how it is the oldest individual among the sample. Whereas least change was observed to be in Ballu, which was the youngest individual among the samples across zoo. The reason for this result could be as young and active individual, it is already comprising of diverse behavior repertoire, having less or negligible room for inclination for such parameters. The enclosure, this slide talks about how the proportion of zone utilization changes at pre-enrichment conditions to enrichment conditions at Delhi Zoo. Basically, the enclosure was divided into 11 zones based on resources and name after features as well as directions. In this plot, we can see the change in utilization as the image on the left is pre-enrichment condition. We can observe that some of the zones are colored black, indicating no usage at all by any by the individuals. Later, this changes with the introduction of enrichment, and the usage becomes more homogeneous as well as number of zones getting utilized by the individuals also increases. Significantly, we can say that there is a positive response of enrichment on zone utilization for each individual. Also, from these heat maps, we can determine which zones were utilized the most by individual bells. In both conditions, the zone cave here, uh, the zone cave here was utilized the most for the individuals Myra, Krishna, and Ballu. In both the condition, we can see the cave is being utilized the most. There are behavior parameters, behavior states which are associated of these zones, and in cave it was mostly stereotypy in the form of pacing. The individual Radha on the other case had high affinity towards right vertex, usually associated with rest behavior in pre-enrichment phase, which changes to a mix of three zones, diluting the effect of single zone of right vertex, ultimately making the usage homogeneous in nature. Similarly, this slide talks about the proportion of zones for Mumbai. The As we can see, tree is the highest maximum zone utilized by the bear Shiva, and the sorry, associated behavior with this zone was rest. For the case of Shivani, the maximum zone utilized by the individual was left grill and associated behavior was head bobbing, which is a form of stereotypy. However, In the condition of pre-enrichment, the second most utilized zone was left ram and the behavior associated with the zone was rest. This slide indicates Shannon evenness index values for each individual for enclosure utilization in pre-enrichment as well as enrichment conditions. This heat map showcased violet as lower values of SEI, whereas yellow being the highest value showcasing even utilization of enclosure, which is an indicator of higher enclosure complexity. The maximum change observed in the value of SEI was seen in Radha rising from 0.36 to 0.82. Hence, we can say it had a strong positive relation with the enrichment. As it is the oldest individual in the sample, a similar trend was also seen in the behavioral diversity, which could be a factor of oldest aged individual benefiting the highest from the enrichment intervention. We can say that enrichment had a positive impact on the site Delhi as it increases in Shannon evenness index values for each individual except for Krishna, where the enclosure utilization declines on the introduction of enrichment. This could be a factor due to a human error where Krishna had an accident in the last 3 days of enrichment phase of the study where an iron door fell on its neck and causing the individual to become highly stressed, altering its natural behavior, increasing the stereotypic pacing, and as we know now, some behaviors are associative of the location in the enclosure, causing a decline in the enclosure usage for the individual, making it more heterogeneous, whereas SE is a measure of evenness. Whereas in the Mumbai, the SE value declined to with the introduction of enrichment in both the webs. This slide talks about how the behavior parameters such as inactivity and stereotypy are being influenced by our covariates. First, I would like to tell you about what is predicted probability. These are typically expressed as decimal values between 0 to 1, where 0 indicates that an event is predicted to be impossible and 1 indicates it is certain to occur. The best fit model utilized for inactivity was interactive effect of treatment as well as zoo, which is site, along with a random effect of individual. These probabilities are adjusted just giving us the result just based on pre-enrichment conditions and when individual equals to 0. And this helps us determine what is the effect of zoo on inactivity alone. The result was marginal different between the zoo for inactivity, showing Delhi as 0.39 and Mumbai as 0.34, to which we can say that Delhi zoo inactivity was marginally higher when compared to Mumbai. In case of stereotypy, the best fit model was an additive effect of zoo and treatment with a random effect of individuals. The predicted probability is much higher for the Delhi group compared to Mumbai group, suggesting a significant difference in likelihood of the outcomes between these two groups. As for effect of enrichment on stereotypy, there was not much change as it doesn't make sense ecologically as an enrichment of 2 weeks can reduce lifelong stereotypic behavior. So the conclusion of the study, we can say that all the sloth bears responded positively to enrichment for behavioral diversity. In case of enclosure utilization, Delhi Zoo showed the enrichment showed positive relation with the enclosure utilization, whereas in Mumbai it showed negative effect. And for the behavior parameters such as stereotypy and inactivity, Delhi was marginally higher compared to Mumbai, and stereotypy levels, Delhi was extremely higher when compared to Mumbai Zoo. I would like to thank these people Thank you. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Sir, one paper talks about this. I cited it, Barrett and et al. 2022. In that, it says, for example, previously I had to calculate it by SPI, but I was not allowed to go inside the enclosure and measure the each zone uh, measures. So that's why. Yes, sir. Yes, sir. Use. Yes, sir. I wanted to see how. Yes, sir. So that I did in my first objective, which was to determine behavior diversity. And for the second objective, I wanted to see how evenly the zones are being utilized. So I calculated, like, I recorded the sighting of the bear in the particular zone, and from that I got the proportion of each zone when compared to whole enclosure, and then I put it into Shannon evenness index. Yeah. Yes, sir. Thanks. Yes, sir. Yes, sir. I have recorded it, but I haven't put into the model. And I haven't- Yeah, see. Yeah. 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 I have provided the same investment at the both sides. It's same. No, sir. The enclosure enrichment varies. Like environmental enrichment varies, where Mumbai has a higher complexity compared to. I have provided pitfall feeders in which you dug up earth and then put bamboo pitfalls, and then you put whatever the feed the zoo is providing, basically fruit, and you hide it, and then. Yes, I have recorded that data, but it didn't. It was not my objective. So also in Mumbai Zoo, it's a soundproof enclosure, so the effect of visitor doesn't really count. But I have calculated for Delhi Zoo. Some of the individual yes. As for example, the individual which are older, they have got habituated, but and they doesn't usually show inactivity, and usually inside the most distal portion from the intruder, from the visitor side. But whereas young individual are not yet habituated. Thank you.
So we had an interesting set of presentations today, but before moving forward, we will break for tea. So I would request everyone to assemble here after 15 minutes, which is at 11:50.
बस क्लिक करते हो
Krishnapriya has a core interest in studying habitat ecology, animal behavior, and evolutionary biology. She looks forward to understanding animal responses in a changing world where human wildlife interface is increasing day by day. All the best, Krishna. Your name.
Thank you, Manus, for the introduction. Thank you, Manus. Uh, Hello, everyone. I'm Krishna Priya. Yes. I'll be discussing about my thesis titled Habitat Selection by Desert Fox in Human Modified Landscape of Thar Desert. Before going to the introduction, I would like all of you to take a look into the diverse habitat type of Thar Desert. One is arid savannas dominated by a species called Vesurus indicus, arid scrublands dominated by Crotalaria buria, unstabilized dunes, and rocky hills. The livelihood of communities living in Thar Desert is majorly pastoralism and agriculture. Uh, the uh, During the past few decades, there was a significant increase in agriculture. The agriculture, in this context, agriculture means seasonal agriculture, which are only active for 4 months, uh, from period from July to November. During the uncultivated season, agriculture looked like this, like kind of barren with few vegetation here and there. Desert National Park was established in 1980 for the protection of arid habitats and its wildlife. It covers an area of 3,162 kilometers, square kilometer, making it the second largest protected area in the country. Although it is such a huge national park, majority of the landscape inside the national park has agriculture and human habitation. Therefore, special structures called enclosures, which are basically fenced habitats, are made for the conservation of Great Indian bustard habitat, which make up to 134 square kilometer out of the 3,162 square kilometer area of the national park. These enclosures are habitat refugia for wildlife in Thar, as well as they act as seed banks for the surrounding areas. My study species is desert fox, Vulpes vulpes pusilla, which is a subspecies of red fox found only in hot deserts of India, Pakistan, Iran, and Iraq. It inhabits arid and semi-arid scrublands and grasslands. The major part of diet come from fruits and insects. This is a video of desert fox foraging. So majority of time when I sighted the foxes, they were actively searching for something in ground. Mostly it will be beetles. Uh, the breeding season of desert fox is from late November, which coincide with my study period. Habitat selection is likely to have an impact on species survival by increasing its reproductive success or decreasing it. It depends upon where the animal chooses to den or nest. For desert fox, according to literature, it is mentioned two type of dens, which are resting dens and breeding dens. The main difference between these two dens are resting dens will only have single opening, while breeding dens, sorry, Resting dens will have single openings as well as multiple openings, while breeding dens always have more than 2 openings. Desert fox always prefer to den on elevated grounds under some plant cover. Anthropogenic activities modify the heterogeneity of natural habitat, and this uh, have an effect on species selecting its habitat for denning. The response to these human modification will be dependent on the species ecology, the extent of habitat change, and also how much the species is adaptable to these changes. Various studies on different species of foxes gave different results. The studies on Indian fox or Bengal fox show high specialization for habitat. They only occur in grassland, while the studies of red foxes show they are uh, selecting grids. They are selecting habitats without such special preferences, and they are also known to survive in human-dominated landscapes. So in the current study, I will be investigating what is the response of desert fox to anthropogenic activities, adding up to the fox ecology literature we know. The objective of my study is to understand the habitat use by desert fox and to examine the effect of protection offered by these enclosures inside Desert National Park. I also intend to characterize the desert fox dens, and another objective is to understand what are the factors influencing den site selection of desert fox in the modified landscape of Thar Desert. My study location is Desert National Park. Since it is a huge area, I selected the northern portion of Desert National Park having more number of enclosures. This is my study area having enclosures as well as area outside the national park for comparison. I will explain my methodologies, analytical methods and results uh, from objective wise. So for objective 1, desert fox occupancy and habitat use, the methodology is followed using occupancy survey. I have designed 3 by 3 square kilometer grid considering the habitat, the core usage of foxes. There is no specific study of core usage of desert fox as such, but there are studies on Bengal fox or other species, and I took literature for getting an idea of their home range or core usage. So it was coming around 3 to 5 square kilometer. So I selected area of 9 square kilometer, which will include any variability. I deployed vehicular transit in these 3 by 3 grids, uh, and I have, for my occupancy survey, I have used these transit for spatial replicate. So the total 6 kilometer transit I have divided into 1 kilometer, uh, 6 1 kilometer spatial replicates, and during this vehicular transit, I have noted down the occurrence of desert fox as well as Bengal fox, because that habitat also have, the landscape also have Bengal fox. Also, while conducting the vehicular transit, I have measured habitat variables such as land cover, substrate, disturbance, food abundance, and vegetation structure in habitat plots, which are 1 kilometer apart from each other. So this is the enlarged version of the habitat plots. In a 200-meter radius plot, I will estimate the dominant land cover type as well as disturbance such as human presence, dog presence, and livestock presence. On either side of the road, I have two belt transect of 25 into 4 square meter, and this is how I quantify the resource variables. In 25 into 4, I walk 25-meter transect, and in a belt of 4 meter, I record the number of burrows of gerbils, the number of burrows of spiny tail lizard. Also, I deploy 1 by 1 square meter transect at the interval of 1.5 meter to quantify grasshoppers and beetles. These species are selected based on the diet studies on desert fox. For my occupancy, I sampled a total area of 369 square kilometer, and I have conducted survey in total 41 3x3 grids. The total number of habitat plots I have was 246, and I did it in 29 days. The total number of grids with fox detection were 12, and majority of the grids with fox detection was coming inside the protected area. The total number of desert fox sighting was 17, and the total number of Bengal fox sighting was 5. In the map provided, I have shown the white stars as Bengal fox occurrence and black stars as desert fox occurrence. So from this you can understand that Bengal fox are restricted inside the enclosures, while desert fox are seen everywhere. Analytical method used. For the analysis, I consolidated the presence absent data I got from spatial replicate as well as the habitat data to a grid level. Also, these are the variables I have summarized. For protected area category, I have two variables which are occurrence of protected area inside the grids as well as distance to enclosure. And for land cover and vegetation category, I have variables such as grassland, scrubland agriculture, barren land and vegetation height. The disturbance factors include dog presence, disturbance here means human disturbance such as human presence or vehicle presence or presence of houses. Also, livestock presence was measured. Resources include food, which is the weighted average of all the food variables I have collected. And I have also measured distance to water. Correlation analysis was done for all these variables. Since my sampling size was less, I couldn't take all these variables for my analysis. So therefore, I performed an exploratory analysis based on univariate GLMs as well as by comparing mean of these variables between grids with fox presence as well as grids without fox presence. From these exploratory analysis, I selected my variables for modeling. I did occupancy modeling. Basically, I modeled detection or non-detection of desert fox on site covariates and survey covariates, which includes all the habitat variables I mentioned before. I did it in occupancy framework provided by M.Sc. I used unmarked package in R. I modeled the detection probability on vegetation height because it was the only variable which could have an effect on the detection of foxes. I modeled the occupancy probability on site covariates such as protection, agriculture, disturbance, food, and compared it with the null model. And the inference was based on information theoretical approach provided by AKK information criteria. I'll be discussing the result as well as here. So based on the analysis, I did detection modeling. So when I modeled vegetation height, it was not showing a significance based on AIC value. Uh, Based on the analysis of based on occupancy modeling, only protection variable influenced fox occupancy. The rest of the variables was not showing significance, which I explained in the table. So here, when you see the effect size, which is basically I have given for each columns of protected area, agriculture, disturbance, and food. You can see only protected area is having a significant effect based on AKK weightage. So it has the highest weightage. Rest of the variables is kind of tricky because sometimes it showed standard error as high as the effect size. That means they have less p-value. The detection probability was 0.06 and the occurrence probability inside PA was 0.62 and outside PA was 0.33, which is shown in the map I have shown before. A majority of the fox sighting was inside the protected area. Moving on to my second objective, which is den site selection, I selected a different study area based on the occupancy result. So for occupancy result, I have occupancy study. I have one study area, and from that I got to know that outside protected area, the grid have chosen has less fox sighting. So in order to maximize the chance of getting more dens, I have selected a different area based on exploratory surveys. So I have surveyed all these areas and I got fox sightings in the regions which I have gridded here. So I had total the total area was divided into 3 by 3 square kilometer grids, and each of these grids was further divided into 36 subgrids for dense search. Dense search was conducted through walking, and for so these subgrid size was 500 into 500 square meter, and this will improves my effort. This will improve my effort for dense search. During this dense search, I have measured direct sightings as well as indirect signs, which I have explained here. So based on when I get a desert fox den, I have noted down what are the indirect signs which are present here. And based on that, dens which have clear entrance, a lot of pug marks, cat, as well as cat of pups, I have considered as active breeding dens. Since the area also have Bengal fox, in order to confirm the identity of these dens, I have deployed camera traps near the den, and based on the capture, I have confirmed the identity. This is the den search summary. I got total 16 active breeding dens. For den habitat sampling, I have created a buffer of 1.5 kilometer around the den and excluded that area, then generated random control points. I have created buffer of 1 kilometer around the den and control points in order to quantify the den habitats. So inside the den habitat, I have taken 5 transect of 50 into 20 square meter belt, and I have measured variables such as land cover, substrate and disturbance in a 200-meter radius from the den or control point. This is how I quantified the resource variable. Additional to the occupancy transect, I have also considered the presence of fruiting trees, which can have an influence on den site selection, because fruits are, like I said, are one of the favorite food species of desert fox. And it was a season where Caparis trees were fruiting. I sampled total 16 dens and 35 control plots, which are distributed in this manner. I used use availability analysis for understanding the den site selection. Habitat variables were summarized in plot level, that is, the transect level data was summarized into plot level. I did correlation and exploratory analysis to narrow down to the few potential variables, because again, in here also I got less sample size, which is 16 fox dens. I compared the habitat characteristics between den and control plots using GLM, and these were the variables I considered. So before GLM, I also did a general linear mixed effect models in order to see whether there is effect of locations on this use versus availability, and this didn't show much effect. Therefore, I selected GLM for my analysis. The best fit model was scrubland, livestock disturbance and fruiting trees. scrubland had a positive effect on den site selection, whereas disturbance such as human presence, vehicle presence, and presence of settlement were having a negative influence on den site selection. Uh, as hypothesized, fruiting trees showed a significant positive relation to the den site selection. Disturbance showed a negative effect. So based on my occupancy analysis, it seems that occupancy was largely determined by the protection. That is, the protected area seems to have a strong influence on the fox occurrence. Other variables didn't show much effect on the fox occurrence. This protected area can offer uh, uh, protection from human activities, especially poaching, and because it provides greater resource in the form of enclosure. So basically, the enclosure had more intact habitat patches as well as water availability. For discussion, den site selection result was desert foxes preferred areas with less livestock and other disturbances, and as well as it preferred to den in areas where there are more fruiting trees. It avoided disturbed areas. It might be the reason uh, that this herders or people used to hunt. Should I conclude? Okay. So my basic question was, how does human modification impact foxes? For that, I did occupancy, I did habitat selection. From that, uh, I got to know that protected area had a significant effect, as I discussed in the discussion part. The main question was whether agriculture has an effect or not. From the use availability analysis, agriculture didn't show an effect because I have modeled agriculture into the general linear model, and it didn't uh, came in the best fit model. But when I looked into the proportion of each land cover types versus its availability, again it was shown that the difference between the use and availability was very less for agricultural land. For scrubland, it was higher. That's why in my study it came that scrubland were slightly preferred more than all the other habitat types. So although land cover didn't show a strong significance, human presence, human footprints and livestock presence showed a strong effect on habitat selection. And grassland were least favored for denning. It might be because from the competition from Bengal foxes which were present highly inside the enclosures, which I couldn't confirm because I didn't get much enough. I got three Bengal fox dens, but that was not enough to quantify that effect. These were the references. I would like to thank these people. Yes. Yes, sir. So initially, when I selected the disturbance variable, dog presence were one of the main variables I wanted to look into. But during my occupancy survey as well as den habitat survey, I didn't get enough presence of dogs in these areas. That's why I couldn't bring effect of these predators in the overall landscape because I compared the landscape inside and outside for occupancy. I didn't get much samples for dogs. So I couldn't say about that.
Actually, a very good presentation. Thanks. Are you the occupancy maps which you have done? Are doing an occupancy survey of 3 by 3 kilometers, 3 by 3 kilometers, and by the dense sites which is different?
The site for occupancy and
Yes.
yes, sir. The sites for occupancy, initially I planned to do dense site selection and occupancy in the same area. But after doing the occupancy, I got to know that the presence of foxes were bare minimum outside in the area I have chosen.
No, I'm saying that when you have done an analysis,
Okay.
when you have selected a subset of the area, do you got dense in some of the areas where your original occupancy had shown zero?
No, sir. Inside the protected area where I had higher fox occurrence, it showed higher than dense also, which is shown in the map.
Can you show the first this one?
Then,
Yeah, this is the den occupancy.
yes, sir. The red triangles are the dens. Yeah. It is similar to the occupancy results. Also. Yes.
But you have not got the den data in the occupancy. You have not got that
No, no,
outside.
no. Both
Because
of
if you have got that in, then you probably have more data. Den is also the presence.
yes.
And then the other question is, when we look occupancy in a continuous distribution, okay, and the assumption of the occupancy is that cells should be statistically independent. The presence of an angle in one cell should not impact the presence in other cell.
Right.
So do you think this 9 kilometer can do that?
Yes, sir. While designing the sampling design, like I took into consideration, took into consider.
What if the home range of the fox is at the corner of the four cells? It's only one fox.
Yes, sir.
How do you take care of that?
Uh, so there is always chances for such occurrence in the borders of the grids, but uh,
Is there a mechanism to correct that?
I'm not sure.
So what you already have done that. You have those smaller grids. The
Yeah.
smaller grids are on how many grids in one?
So subgrids were only for dense search.
You have selected fox presence.
Yes.
Yes. So just run your occupancy on the smaller grid size.
Okay.
And that is because smaller size probably will give the statistical independence in the larger ones.
Okay.
And that probably will give you a far better results than running on a 3 by 3 spectrum.
Okay, sir. I'll do.
Just in continuation of the question, for the grid size, you are choosing 3 by 3, while 1 meter for the occupancy as well as the tensile selection.
Yeah.
So, what is the basis for choosing the grid size?
Yes. The occupancy framework uh, provided by McKinsey uh, comes with the assumption of taking large enough area which will incorporate home ranges of multiple individuals. So from the literature study, I got to know that the average home range of desert fox, not home range, the core usage of desert fox was 2 to 3 square kilometer. So by taking an area of 9 square kilometer, I can incorporate that factor. I took a large enough, I could have taken 2 by 2 also, but I took it like 3 by 3 because I didn't know about this particular species as such. I was referring on literature. So in order to consider that, I took a larger area.
Good. And another question is that during your tensile selection survey, have you observed any other cohabiting species or coexisting species in the den? Is there any interaction positive or negative?
Okay, inside the den.
So you could have done the site survey in the entrance of the den,
Yes,
right?
yes,
To
I have.
understand the fox presence.
Yes.
So is there any other sign have you got in the den?
No, sir, because the other animals which use dens in that landscape, one is Bengal fox, which definitely won't use an area where desert fox is found. And other animals such as reptiles and gerbils won't prefer to stay near the den since they are the prey species of the foxes. So I didn't find any such exhibitions.
Based on your research that you have mentioned, so you have mentioned that within the protected area, the rents were higher. I mean, the population is good comparatively outside the protected area. So, what would be the reason? You have mentioned also the hunting pressure is there in the outside, or is there any food availability is less compared with the within the protected areas? Or have you
Yes.
compared with those regions?
Yes, sir.
There are
So
other questions waiting. You can discuss that later. Yeah? Sorry.
No problem. Yeah, so we have two questions by Dr. Hunter. The first one is, how many
Take
desert
only one is allowed.
only one. The first one is only one is how many desert fox and red fox dens will you find? What were the structural differences between both fox dens that you observed in your field?
so desert fox dens and Bengal fox dens.
Yeah, she meant Bengal.
Okay. So I got total at the end of my den search, I actually got 70 fox dens, which included resting dens as well as breeding dens. So based on the methodology I have explained, I narrowed down it to 16 dens.
Okay, done. Last question.
Sample a lot of food resources of foxes, right?
Yes.
What were the key patterns coming out of that?
Yeah, so that was one nuances I had in my study. That even though I have surveyed extensively for food resources, majority of the data was coming as 0 0, like zeros were a lot. So that explained the heterogeneity of the habitat in the desert. So in order to accommodate such heterogeneities, I need larger transects or larger belt transect. Then I might get more abundance of resources, and then I'll get to know the result. Thank you.
Moving on to desert mountains, we have the final presentation by Prakriti, who is exploring ecological separation between Himalayan blue sheep and Himalayan ibex during the harsh winters in Trans-Himalayas. Prakriti is interested in studying species interactions and their responses to environmental changes, and she is particularly fond of high altitude ranges. All the best, Prakriti. Over to you.
Good morning, everyone. I'll start with the background. Uh, so, according to resource selection theory, two similar species occupying the same area and utilizing the same resources, they coexist only when they segregate in terms of habitat or food preference or both. So, competition occurs when the resources are limited. And there are two types of competition possible: interference competition and exploitative competition. In interference competition, the individuals they directly interact in form of aggression and territorial disputes, and in exploitative competition, the individuals they indirectly compete for the same resources, where one species exploit the resources by more consumption and decreasing the availability for the other species. So, since my study was carried out in Himalayan region, Trans-Himalayan region, Himalayan ibex and Himalayan blue sheep are the two major prey species for the snow leopard. So, Himalayan ibex being the large mountain goat and well adapted for climbing, and Himalayan blue sheep is a medium-sized ungulate which has both goat and sheep-like characteristics. So, ibex is distributed in the rugged terrain in the high altitude region. Blue sheep is distributed in the Tibetan plateau. Given they also overlap in some areas of China and in Ladakh region, and also in regions of Khyber and along the Pir Panjal range in Himachal Pradesh. So, given since both the species are similar, given the overlap, so I wanted to, I mean, how are they? I wanted to look how are they separating in terms of space, habitat, and diet. So hence my title, Exploring Ecological Separation between Himalayan Blue Sheep and Himalayan Ibex during winter in Indian Trans-Himalaya. So studies have indicated that livestock outcompete wild herbivores, and there is also segregation among the wild herbivores. For example, in a study on Tibetan Argali and blue sheep, which indicated that they segregate in terms of habitat, aligning with their anti-predator strategies, and one study has looked into broad habitat overlap between habitat segregation between ibex and blue sheep, and my study will look into habitat segregation at finer scale. My objectives are to assess the patterns of habitat selection and how they differ in the use of space. Since the research was carried out during winter, snow factor is a major important and also limiting factor. So I wanted to look how snow cover, snow depth, and proximity of to escape terrain influence the habitat selection by both species. And to evaluate, the second objective is to evaluate the niche overlap and how similar or dissimilar is the selection and utilization of forage and their temporal overlap. So the study was carried out in Trans-Himalayas in Spiti Valley, in the which lies in Lahore and Spiti district. So it was carried out in Kibba Wildlife Sanctuary, and the intensive study area is 57 kilometers square, where the elevation ranges from 3,600 to 5,200, and the vegetation type is alpine scrub. And there is diverse fauna in my study region with two apex predators, snow leopard and Himalayan wolf, and the inhabitants follow Tibetan Buddhism. And this is the general design of my study. So I have divided the study area into 500 by 500 meter grids, and I chose monitoring trails and vantage points for sampling. And for my first objective, I laid 30-meter radius plot and collected variables like distance to cliff, cover values, snow depth, etc. And also topographic variables like elevation, slope, aspect were extracted from DEM data. And I also placed camera traps to assess the temporal activity pattern. And since escape cover is an important aspect for the herbivores, so I considered number of directions in which escape terrain is available. So I considered 8 directions. So the basic concept is, since snow leopard is a solitary and a stalking predator, so if it blocks one escape route, the animal will have other 5 escape routes to escape. And since I wanted to look how these ungulates utilize the terrain type in my study area, so I categorized them into interspersed rocky slopes, cliffs, free, rocky slabs, rocky slopes, smooth slopes. And for analysis, I did Jaccard similarity index to assess the spatial overlap, and I did NMDS to reduce the dimensions of habitat variables and to look into the segregation of ibex and blue sheep, and I used logistic regression to see what are the factors that are affecting the use habitat selection of these species. So for my results, according to Jaccard similarity index, 17% of the grids were shared by ibex and blue sheep, where blue sheep occupied spatially distinct zones in Dumlane, Getten, Kibber, Cheecham areas, whereas ibex used only Cheecham and small part of Kibber. Um, and and for habitat utilization pattern, this graph clearly depicts that there is segregation in blue sheep and ibex use. So the axis one represent. The loadings are slope and snow cover, and for the axis two, it is distance to cliff and snow depth. So the availability points are coded in red, and blue sheep use points are coded in green, and ibex use points are coded in blue. So from this, we can infer that blue sheep uses broad range of habitats that are available. ibex is restricted to a smaller region, so which indicates it has specific habitat requirement. Uh, and I and when I ran logistic regression, so ibex the variables um, that influence their habitat selection is elevation and its quadratic term, number of escape terrain directions, and terrain type. I have considered quadratic term because not all variables will have linear effect. So to see whether there is a peak at intermediate levels, I use this term. And interspersed rocky slopes is more utilized, and scree and smooth slopes are less utilized, with no utilization in valley bottom. And for blue sheep, it is slope, vegetation cover, distance to cliff, and number of escape terrain directions. Although distance to cliff and vegetation cover did not have a significant effect. And since my research question was to look into how snow cover, snow depth, and distance to cliff affect their habitat selection, so snow cover has a significant effect on ibex use, and distance to cliff has a significant effect on ibex use, whereas others don't for blue sheep. So summary is, despite the limited forage availability in my study area, there is minimal spatial overlap, and both the species they avoid deep snow areas, with ibex using more snow cover areas compared to blue sheep. And there are studies that have indicated that ibex are said to dig out snow craters and feed on the vegetation, while the blue sheep they restrain from that. And ibex uses interspersed rocky slopes. It requires multiple escape routes and close proximity to cliffs. That is why they were using. They had that specific habitat requirement in that. And blue sheep it uses steep slopes with high vegetation cover and feeding. It can feed away from cliffs and can retreat to cliffs once they detect the danger, as they can run down the slopes. And for my objective two, I laid a 10-meter radius vegetation plot. In that, I laid 1 by 1 meter quadrats randomly, and I collected these variables, and also collected pellets after when I saw when I saw the animals defecating. So the pellets were oven dried, then powdered, and then slices were prepared, and then uh, looked under microscope to determine the proportion of monocot and dicot. And for analysis, Twinspan was used to get the vegetation clusters, and to compare the use and availability for vegetation communities and monocot and dicot, I used Evelef selectivity index, and I used overlap packets to assess the activity pattern and temporal overlap. And the results are uh, I got our 8 different clusters were obtained from the Twinspan analysis. The species here is Aquilegia and Nepeta, and here it is Cousinia and Potentiala. Here it is Crastenikovia and Polygonum. Here it is Elymus and Piptathirum. This is Carex dominated community, and Caragana and Ephedra dominated community. Rosa Webiana, C. Buckthorn. And for ibex, they preferred Rosa Webiana and Seabuckthorn communities more because these communities were prominent in ibex use areas. Blue sheep preferred Caragana ephedra and high vegetation cover communities like Elymus species Piptathirum and Crastenikovia, Cristolia and Polygonum species community. Uh, so both the species had higher dicot proportions in their diet, although the availability of monocots were more compared to dicots. Dicots were utilized more than availability. And both the species showed unimodal pattern. The ibex was active showed peak activity period during 10 o'clock to 11 o'clock hours, and ibex from 2 o'clock to 3 o'clock. And there is 73% overlap in their activity pattern, which indicates that they are active in I mean during the daytime, and they overlap. And summary, blue sheep use Caragana ephedra communities more because this community is widespread in my study area because of high livestock pressure, and both consume more digots than monocots, which is consistent with other studies which indicated the seasonal dietary shift from monocots to digots from summer to winter. And there is substantial overlap in their activity pattern. So the findings are ibex and blue sheep, they segregate in terms of habitat use and space, and dicots form the major portion of diet. And ibex trades off food for security. Um, it, um, it prefers rocky terrain um, for escape, and also even though it has sparse vegetation, and prefers escape terrain available like within 100 meter, and also high snow cover areas compared to blue sheep. Whereas blue sheep, they balance both food and security. They can run down the slopes, and they can retreat to the cliffs when seeing danger. So I would like to acknowledge these people, and these are my references. Thank you.
Faculty, do you think there is any publication from India which talk about escape terrain for blue sheep and ibex, especially for blue sheep?
Yes, sir.
You have not referred them?
I have referred them, but
Yes, I was not because I see all over literature which is 10, 15-year-old, but not a recent one. Just published some of years ago. But the findings are all the same. So don't worry.
yeah. So, but when I compare both ibex and blue sheep, so when I compare, then I'm telling that ibex uses more rockier terrain compared to blue sheep. Even though blue sheep also uses cliff because they are more related to goats, but they maintain some distance since they can run down the slopes and go to the cliff because they need high vegetation cover also. Blue sheep needs, whereas ibex, they can manage with sparse vegetation, but they need security. Security is more important for them. Yes, sir.
Activity pattern.
Yes, sir.
Do you think wind chill and wind speed in the day have any effect? Have you observed anything when you were there? Wind chill and wind speed varies across the day.
Yes, sir. It varies across the day, and the thing is, this. I mean, with this temporal overlap, I have fewer detections in my camera tab because of winter conditions and all. Due to low monitoring, I've got less detections, and because of that, I've got this data. But yeah, there was this effect of wind speed and this one, but what I have observed with ibexes, they go near the cliff because there the wind speed reduces because so they okay. Uh,
Till the wind speed comes down, then try to exploit. Whereas blue sheep comes earlier down. So if you have
yes.
a camera that's if you put a camera that's very close to it, you will see them resting there.
Yes. Thank you, sir.
Just one clarification. So for the ibex, you have mentioned that they are specific to the particular habitat where high snow cover area is there.
And also,
So if you are looking at the spatial overlap between the ibex and the blue sheep, wherever the ibex is there, the blue sheep, the diversity level is very minimal. So that is because the blue sheep is not preferring where the ibex is there, or is there any other reason?
uh, if I can show you
Yes,
the
please. Go to the
yeah. So here, so in that area where both ibex and blue sheep uses, there the terrain is not very rugged. If I show that photo of okay, one second. If I show the photo here, this kind of terrain is available where the ibex was using. So where I mean blue sheep cannot use this kind of terrain because this is too rocky and it has sparse vegetation. So that is why ibex were there restricted there. Although some space, I mean both the species overlap, but the terrain is not rugged like this. It is like normal cliffy like area. So it's like
Mostly to do with the terrain, right?
yes, sir. Yeah. Thank you all.
So with this, we conclude today's session. Tomorrow's session will